Thank you, sir.
I didn't say anything about your pants.
You didn't say anything about my pants. Good. So, okay. So today I'm going to give you some examples of what insights we're gaining into the heterogeneity of B-cell responses by looking at vaccination and also by HIV infection. Just to start off, and I know that you've heard all this morning already. How do we use this? Oh, there we go. But this is the initiation and recall of recall B-cell responses. And clearly this is a simplified version of a germinal center, and where the main players are the B cells, the FTC that presents the antigen to B cells, and the CD4 TFH, which you've heard a lot about today. So the CD4 TFH are very important in this context, as once the antigen gets presented to the B cells, they then require CD4 T cell help and move to the dark zone. If they don't get CD4 T cell help, they just apoptose. Once in the dark zone, they undergo somatic hypermutation and proliferation. They then migrate back into the light zone, where their fate is then decided. They either go to be short-term and long-lived plasma cells, which then contribute to circulating serum antibody in the bone marrow, or they populate the peripheral blood of a heterogeneous population of circulating memory B cells, with greater diversity actually than in their BCR than the antibody pool. And this is essentially what we call recall responses. And these are high affinity memory B cells. And upon re-encounter with their antigen, they can actually go back into a GC or form a new GC and then start the whole process again of somatic hypermutation. And this is, we could view vaccine success as this process, as this cyclical process. So the study of these plasma cells that are released from the lymph node, we can obtain IG sequences. Am I hitting something here? We can obtain IG sequences from the plasmablasts, and this would inform vaccine design. For example, I know some of you are doing this for a dengue vaccination right now, where you obtain plasmablasts at 7 to 10 days post-boost. And these would better represent circulating antibodies. So you can compare this to the B-cell receptors from this compartment and have a look to see which the familial relationships and the clonal relatives to serum antibodies. We can also identify therapeutic antibodies in this way. We can study the memory B-cell pool where we obtain IG sequences exactly the same way. And we can characterize the potential antigen-specific recall B-cell responses. And we can identify therapeutic antibodies, for example, VRC01 for HIV infection. So our interest really lies in the assessment and the evaluation of frequency and quality of antigen-specific B cells following vaccination, determine the recall potential by sequencing the BCR repertoire, and we can also track B cells in the lymph node compartment during vaccination or indeed infection, like with HIV, which I'll go on and explain. So to assess the frequency and quality of antigen-specific B cells, we've been developing various probes, and I'll go into how we develop the influenza probe. And then we can evaluate the activation states and the transcriptome profiles of antigen-specific B cells. So back when I first started at the VRC, we were trying to understand a vaccine trial, VRC 310, which was a vaccination with H5N1. However, we did not have an antigen to pull out our B cells with. And so this is an HA. And if you just use regular HA like you would find on the surface of a virion, what you actually get is you stain every single B cell, because every single B cell has sialic acid on it. However, if we mutated tyrosine at position 98 and we change that to a phenylalanine, then we can abrogate sialic acid binding completely, and these cells now represent the antigen-specific cells. once we have a probe, be it a probe for influenza or for HIV or for tetanus toxoid or for human papillomavirus, we can do a number of things. So here we have an H1 probe and an H5 probe, and this is pre-vaccination and post-vaccination. And as you can see, these populations have increased. So we can do sequence analysis, we can do B-cell maturation analysis, we can isolate monoclonal antibodies, we can have a look at the kinetics of the memory B cells, we can look at transcriptomics, and we can look at B-cell phenotyping. So we do have, and we've developed a 15-color panel for looking at different subsets within the periphery of any individual. And so we can actually identify these 15 different subsets, and then when we combine them with specific probes, such as HIV GP140 or the influenza probe I've just talked about, it actually becomes really a powerful tool. So the resting and activated memory B cells are very important in recall responses. For example, a vaccine boost, or even when we deal with virus variants, such as in HIV or Hep C. If we use CD21 and CD27, we can split the B cell population up into intermediate memory, resting memory, activated memory, and tissue-like memory. The resting memory is critical for maintaining the recall antigens, and these populations are reduced in HIV-infected individuals. The activated memory, there's some evidence to say that these are the ones that traffic to the lymph node. There's an increased level of Ki67. There's increased population shortly after vaccination. They are prone to fast-mediated apoptosis, and they're associated with HIV-induced immune activation. The tissue-like memory, exhausted or atypical cells, which are CD21 negative, CD27 negative, form a very interesting population. They have increased levels of inhibitory receptors. They have altered expression of homing receptors. They have stunted somatic hypermutation and replication. And these are actually enriched in HIV-specific B-cell responses because the characteristic of chronic infections such as HIV, HCV, malaria, and also some autoimmune disease. This is again another plot of CD21 against CD27, and this is from a recently vaccinated individual. And you see there's a lot of cells landing in this activated memory phenotype. One of the questions we want to ask is, what are the phenotypic signatures and lineage relationships between the resting population and the activated memory population? Indeed, we can also have a look at the tissue-like memory, or the exhausted cells, and see if they can recycle, or is this just a trash can for all the B cells? What is the clonal relationship with resting and activated memory? And then is this affected by the vaccine boost regime? So we can ask these questions. And here we've used the H5 vaccine trial once again. And we've had a look at the H5 specific B cells versus the H1 specific B cells. And this is at 2 weeks post-vaccination. And what we can see is that you have a decrease compared to the H1 B cells, a decrease in CXCR5. You have an increase in CD22, and also an increase in these markers, which are markers of activation. If you actually have a look at this, and you just have a look at the CXCR5 and CD22 populations that came up in this analysis, we see that at 2 weeks, the population segregates out into those that are CXCR5 high and those that are CXCR5 low. And then if we have a look at if we then plot them against CD21 and 27, we see that the activated memory population is actually enriched for antigen-specific cells at this time point. So the H5 specific B cells demonstrate a more activated phenotype compared with the H1 specific B cells, and they're quite closely related. So new and developing B-cell responses following vaccine and boost fall into this population here. And this is very useful for immune monitoring and also to directly sort and analyze B-cell recall or the activated pool. Here we have the same plot, but this time what we've done, we've taken an individual. This time this individual was vaccinated as part of an HVTN trial. So this is an HIV vaccine trial. And this sample was taken 4 weeks post-boost. And these are all probe-specific B cells, and we've sorted these for resting memory, activated memory, tissue-like memory, and for plasmablasts as well. Each one of these dots represents a cell that we sorted into an individual well of a plate by a method called index sorting. We can then take that cell and we can do phenotypic profiling, courtesy of how we sorted it. We can do lineage relationships by extracting the cDNA and having a look at the immunoglobulin locus. And we can actually have a look at some transcriptional profiling. And I'll go on and I'll show you some evidence of this. So using just 96 genes in a fluidigm biomarker experiment, we can actually segregate out plasmablasts and plasma cells, resting memory, and activated memory. A better representation of this is if we do a discrimination analysis, and we see that the plasmablasts are very, very different from the activated, resting, or the tissue-like memory cells. And these are some of the genes. These are the top 10 genes that are associated with this separation. By doing this and by including different genes, we can actually expand the flow cytometry panel, so we can do this iteratively, and then we can actually get a panel that's specific for plasmablasts. We can also have a look at the resting memory, the activated memory, and tissue-like memory. And what you immediately see from this plot is that the tissue-like memory and the activated memory are actually they're very much related. Whereas the resting memory does form a distinctive population. And if we have a look at the cells that are responsible for taking this resting memory population away from the other two populations, it's involved in trafficking. There's some inhibitory receptors in there, but mostly trafficking markers are downregulated compared to the activated memory. So we can use this method, as I said, we can use this methodology to iteratively expand our flow panels and better resolve antigen-specific memory B cells, or even guide RNA-seq analysis. So in summary for this part, development of more specific B-cell probes enable identification and understanding of the kinetics and the role of activated B cells that make up the recall pool. Do all antigen-specific B cells follow similar phenotypic and transcriptional pathways? So what we're doing with this, we're now using different probes and looking at different pathogens to see if the B cell responses are exactly the same. So do they follow A, B, C, or do they go A, D, X? Likewise, if we have a look at vaccine versus infection, or even different vaccine regimen. We can also have a look at HIV, tetanus toxoid, influenza, and the list goes on. So this also informs the relationships of recall memory B cells with plasma cells and serum antibody. So if we have a look at the BCR sequences in a flu vaccine trial, we can answer some very important questions, such as why do current influenza vaccines elicit poor cross-protection? For those of you not familiar with influenza, influenza falls into two groups. This is group 2 up here, H3, H7 you may have heard of, and group 1, which is H1, H5, and H2, which are common. So H1 is very common in circulation. The reason why they are very diverse, they have antigenic drift and antigenic shift. Just to go into that a little bit more closely, antigenic drift is the gradual change in the hemagglutinin. That's accumulation of point mutations, and this accounts for seasonal variations. This is essentially why we have to be vaccinated each year. Then we have antigenic shift, and this is a rapid change or a recombination or reassortment. For example, H5N1 was an avian reassortment, and H1 and pandemic H1N1 was a reassortment from an H1N1 from a pig. And clearly we know a lot about pandemic H1N1 because in 2009 we just got over a pandemic where lots of people died. We have a look at the molecule of HA itself. It's made up of HA1 and HA2, but in general we can divide it into head and stem. Now, the head-directed antibodies have high potency but very little breadth, again, why we have to get vaccinated each year. and they dominate the immune response from natural infection and annual vaccination. However, STEM is more conserved, but it's seemingly only targeted by very few antibodies, and they're quite rare, and mostly very similar. Antibodies directed towards STEM, and if we can work out how we can boost antibodies that are directed towards STEM, this would make up a universal influenza vaccine. So rather than us getting vaccinated every year, we would just get vaccinated once. However, there is a competition for the B cells. There is a competition with head-specific B cells, and I'll go into that a little bit later. So if we use our flu-specific probes again to characterize the B cells, and we use an H1 probe, and we use an H5 probe, we see that there's a population of H1 that increases, and you see that we can detect, and we see that this doesn't really increase throughout the H5 trial. There's a population of H5 only specific cells, and these do increase because it's a neoantigen. However, we also find this cross-reactive population, which we found was directed towards STEM. And because H1 and H5 are both part of group 1, these STEM reactivity are group 1 specific STEM reactivity. And these, oddly enough, don't really change in frequency either. So what's going on? So H5 B cells undergo classic primary response, class switch, and expansion. Closely related H1 does not undergo similar expansion, as we can see here. However, re-expansion of existing switched H1, H5 cross-reactive cells that are directed towards STEM are expanded slightly, but then there is also at the same time, there's an increase in serum antibody towards STEM, which then drops off pretty quickly. So if we go into those STEM-specific cells and we look at the single B cell and we take out their immunoglobulin and we have a look at the relatedness of all the immunoglobulins within that population, we can do this by this single cell sorting again. We can generate cDNA, and then we can use nested PCR primers to amplify the heavy and the light chains. For the next couple of slides, I'm going to represent the antibody relationships in a pie chart, which Rick loves, right? Rick loves pie charts. So in the center figure is going to be the number of clones that we got from a certain individual. Each pie is a unique clonal family, and then the size of the pie is the frequency of that clone that we found within the population. So within this population or within this population. And what we find is that the H1 positive, H5 positive, or STEM B cells actually are dominated by one heavy chain, which is VH169. And these are all different clonal families, but they're all using this one heavy chain. Whereas if we have a look at the H5 population, you don't see that dominant heavy chain at all. So the H5, H1 population is relatively high, but it's transient B cell frequency. They have a dominant BCR heavy chain usage of VH169. These are specific just for group 1 viruses. They expand in vivo in response to heterologous influenza exposure, such as the pandemic H1 or the H5N1. So what about B cell responses, which is the vaccine question, right? What about B cell responses that bridge that gap between group 1 and group 2? So if we use, rather than using an H1, H5, we use an H3, which is a group 2, and we use an H5, we still find those cross-reactive stem cells, but they're of far lower frequency. Now, the homology of this is 34% homology in the head and 54% homology in the stem. So can we pull out B cells specific for group 2? Well, it turns out that we can, but once again, they're dominated by certain heavy chains. So there's a large clonal expansions within H3, H5, with common BCR sequences, such as VH118, 430, and 61. Now, these are coming up in different individuals. So different individuals are using the same heavy chain. So it's rather like public and private clonotypes in T cells. Indeed, if we have a look at a certain heavy chain and then we mutate it all the way back to the unmutated common ancestor, we find that there's a number of different families or a number of different branches of the same family that are in circulation constantly. But we don't have circulating stem antibody that will protect us from different flu. So these sequences are obtained from the H1H5 stems, cross-sectional sampling, so we have 102 heavy chains. The hydrophobic, the CDRH3s are around about 21 amino acids. The somatic hypermutation is anywhere between 2 and 13%. They do undergo affinity maturation, clearly, because it's 13%. But there's only a modest increase in both binding affinity and neutralization potential. So why do some of us have potent anti-influenza stem BCRs, yet we remain susceptible to infection? So I propose this model that this is head and this is stem. Okay, so again, we'll go back to our dynamic GC again. And the CD4 T follicular helper cells seem to be all against the head. We haven't found any CD4 T cells that directed against the stem. So the stem-specific B cell responses are using CD4 help from the head. Thus, they get selected against. So head-specific plasma cells, there's few stem, but there's a lot of head here, and they go off and they go to the bone marrow, and they go into the circulation. So you have circulating antibody. Whereas short-term plasma cells, upon encounter of antigen, so for example, if we do vaccinate with an H7 or an H5, we'll boost this population into, and they'll become activated, and then the activated ones will become short-term plasmablasts, and those short-term plasmablasts will release antibody. And so we'll have a transient antibody in circulation. But these cells are never going down this pathway, they're going through this pathway. This was also observed by Antonio Lanzavecchia, who isolated a group 1, a very potent group 1, group 2 specific antibody called FI6, which is directed towards stem round about here. And he only ever found that antibody from doing plasmablasts. He never found that antibody from doing memory B cells. But he found lots of clonal relatives of this very specific antibody, but they were never as potent. So we need to acquire more knowledge about governing the selection and the fate of naive plasma cells and plasmablasts and memory B cells and the lymph node, and we need to study the lymph nodes in particular, and the relationship between the memory B cells re-entering the germinal center and which TFHs they actually use. Novel prime and boost strategies for influenza. For example, if we prime with full-length head and stem, and then come back and we boost with stem. We're trying to work out some of these actual vaccine regimen now. And we can do this in NHP, in very controlled studies. And this does have implications for the design of new imaging for HIV. So BG505 SOCIP is, there's a lot of work going on to this in this BG505 as a potential imaging for HIV because it binds all of the neutralizing antibodies. But there's no guarantee that we're going to obtain the neutralizing antibodies because we may just get B cell responses against the whole of the molecule and they don't go to being plasmablasts. So the final part of the talk is how we're tracking cells in the lymph node during infection. And we're doing this in collaboration with Susan Moore, who's over in Tony Fauci's group. So by using CD38 and IgD, we can actually recognize 4 different subsets from the lymph node. We can recognize naive cells, which are IgG positive. We can recognize memory B cells, which are IgD negative and 38 low. We can recognize germinal center B cells, which are 38 high and IgD negative, and plasma cells, which are very high for 38. And we can segregate out these different populations. And by the use of probes again, we can use a GP140 probe, and we see that within this population, there's a lot of antigen-specific cells coming out of the GC-specific. Whereas in the memory B cells, we have less of a frequency against GP140. However, if we use a GP140 CD4 binding site probe, we can obtain these populations and have a look at the relationships in the different compartments. So if we look at again, this is the pie chart, this is the number of clones that we obtained from this one individual. This is an HIV viremic individual, and we use the CD4 binding site probe. What we can find is that in the GC, obviously, as we have heard all morning, you have lots and lots and lots of different clones. Which ones get selected and then which ones leave the GC to go into the lymph node? We get an insight into this here. So like one family here, and I'll go into this in a second, one family here is expanded in the lymph node memory B cells. And obviously, the amount of somatic hypermutation that's seen in the GC is very different to that seen in the memory population. Once we get out into the periphery, where we can use the same probes and have a look in the periphery, we can see that these small populations are very much expanded. So a population that is only 0.74% of the B cells that we could obtain then becomes nearly 10% of the B cells that are in the lymph node memory, and then becomes 35% of the B cells against this particular probe that we find in the periphery. So these frequencies are not surprising from what we know about the GC. But this is a good way to actually track which B cells actually then go on to produce plasma cells and go into long-lived memory, and which ones go into form the memory B cell pool for recall. If we go back and we think about what's the phenotype of those cells, well, I told you that there was an accumulation of tissue-like memory in HIV-positive individuals. This is actually not that big a population. So if we take this same lineage, the SM479, they mostly fall into the activated and resting memory populations. Clearly, this is very interesting from a transcriptomics point of view, and so we're actually following this up right now. And we're also following up using V1, V2, and EMPA probes to do a similar analysis from the lymph node. But we do have to follow up on using plasma blasts. The problem with that is that the plasma cells downregulate their B cell receptor. So it's very difficult to pull out antigen-specific plasma cells. In order to do this, we have to use some tricks, and we're actually trying to work out some methodologies now to try and obtain the same B cells for V1, V2, or EMPA, or for CD4 binding site from the lymph node. We're also synthesizing the BCR sequences that we found and having a look at the specificity and the affinity of those B cells. So low frequency of SM479 sequences are present in the tissue-like memory. This was actually a surprising finding. We thought that it'd be a lot higher, because in malaria, what they've actually found is that most of the malaria-specific B cells fall into that exhausted or tissue-like memory CD21, CD27 negative population. And another thing that Susan's observed is that the amount of somatic hypermutation in the tissue-like memories versus the activated memory or the resting memory populations is very much reduced as well. So we need to do some direct sequencing on the plasma cells, so maybe next year I'll tell you some more about that story. So the overall summary is that memory B cells serve as a backup for long-lived plasma cells. There's a broader BCR repertoire in circulation, and it's a rich source of data for vaccine efficacy, because it's basically a record of absolutely every antigen that we've seen. And I think this is probably adapted because of variation in pathogens such as viruses. However, vaccine design can be confounded, as I showed you with the influenza, by selection of events determining the fate of cells that exit the GC. So it's the B cell recall pool or the long-lived plasma cell, and the long-lived plasma cells clearly contribute to serum antibody. So the phenotypic analysis of recall memory B-cell responses are important for immune monitoring and vaccine design. And we look forward to better probe design, and also we can assess adjuvants and boost regimen by using these probes. So I'll end there, and I want to thank Adam Wheatley, who did the vast majority of this work or contributed in some way, Sarah Andrews and Madhu, and also Sandeep and Michael in my group. I want to thank Rodrigo for helping us out with the three-way profiling, and Rick. Mario and Prateep have been invaluable. And then, of course, the study volunteers from HVTN 204 and 310. And also, finally, if you're looking for a postdoc and you like some of the work that we're doing, this is my email address. Feel free to drop me a line. Okay, thank you very much. I'll take some questions.